my great pleasure to introduce Jared Freedman for his PhD defense talk. Jared came to me, kind of approached the lab, just having finished it in Master College, having done a really cool undergrad advanced project looking at the evolutionary genetics of tropical frogs. So he came in with really good molecular bench skills, a lot of ideas about what he would do in the lab. And for those of you familiar with our lab, you might know we do a lot of different things, but we've always had one thread using molecular genetic techniques to understand how populations are connected across these big desert river landscapes. And in the past, we've done that with just sequencing single specimens with mitochondrial DNA, developing microsatellite markers to get a bigger picture, then using SNPs and 2D rat types of approaches to kind of get a bigger slice of the genome. And we made a lot of progress looking at a couple taxa at a time. We did 3 taxa at a time across these landscapes and understanding how those populations are connected and how they're diversifying and so on and so forth. Well, right off the bat, Jared suggested, hey, why not eDNA, environmental DNA? And I was like, man, I've just seen so many difficulties people were having with it methodologically. Like you had to prove something in terms of the methods in order to get it to go forward. No. Well, Jared looked at it and really read through the literature and found some very promising papers that were inspiring. And so I was like, okay, let's do this. Let's try it out in the Grand Canyon ecosystem, which is where we've been doing a lot of projects at the time. And since then, Jared has just blown the lid off of that method in terms of how our lab would do it. And I've fully embraced it, and it's thanks to this person. And I always say I'm not going to steal your thunder. I think I said that for the last few minutes too, but I can't help but give you a little preview. So there are 2 really important questions that Jared tackled in his work, and he's going to talk about to some degree here. And one is a management question. So the Grand Canyon ecosystem, huge iconic river, lots of threatened and endangered fish, but the river itself is so messed up by flow regime changes due to dam operations and reservoirs and other ecosystem factors that it's a problem for the fish. The food base of aquatic invertebrates there is very, very depauperate. And people have always wondered, well, there are all these tributaries coming in, because could those tributaries supply the mainstem Grand Canyon with kind of fresh recruits of aquatic invertebrates? And we've been trying to answer that for years, and I think Jared has pretty much nailed it. The answer is a resounding no, at least right now, the way the ecosystem is managed. We'll show you the details. The second thing is, I think, a real big frontier in how we're going to do population genetics going forward. So I mentioned that a whole PhD would be developing molecular tools to look at the dispersalability of like maybe 3 species at a time. And that's a lot of development of the techniques, a lot of taking tissue samples, a lot of sequencing, so on and so forth. What took a PhD, an entire PhD to do for 3 taxa, Jared has now done in one swoop for well over 100 taxa at once using eDNA as a method. And I think this is going to be an exciting new kind of methodological push forward for us and some other labs that are interested in these kinds of questions. So with that, I'm sorry to see Jared finishing up here, because I feel like we're just getting started, but we have some really great papers we're going to put up together and hopefully get all that done before he starts a postdoc at Cornell University in the fall. So with that, Jared, take it away.
Thanks for a great introduction, Dave. I pretty much don't need to say anything now. I think you covered all of the important parts. I guess I shouldn't stand in front of the screen here. Yeah, so today I'm going to be sharing some of my research for my PhD, looking at biodiversity patterns in the lower Colorado River Basin using eDNA, ranging from populations up to ecosystems. So Oh, that's not working now. There we go. So starting off, who here's been to the Grand Canyon? And a lot of you have been to the Grand Canyon. How many of you have been down to the river of the Grand Canyon? Still a decent amount of you. So those of you who have, or who just listened to Dave's explanation of the system, probably have some appreciation for the amount of landscape variability that's found in the Grand Canyon. So there's a lot, especially in the aquatic habitats, ranging way up high on the rim, all the way down to the big Colorado River down at the bottom. And so the big overarching question that I was interested in addressing with my PhD is, how does the landscape influence the distribution of biodiversity kind of throughout all these ecosystems? And to do this, I looked at several different measures of distance, so at different spatial scales, spatial distance, environmental distances, which are essentially the variation in environmental characteristics between habitats, and then biological distances, so species-specific interactions with the landscape. So aquatic habitats are important ecosystems, and landscape distances are really relevant for studying them because aquatic habitats are really sparse on the landscape. Water is pretty rare, and it provides a unique habitat type. So habitats within a river network are linked by their hydrological connectivity, how the rivers flow to one another. So following down one branch of the river up to another to connect habitats. They're also connected by their relationship to one another across terrestrial distance as just standalone patches on the landscape. And this creates a dual spatial representation of the landscape where different ecological processes in rivers are governed by different types of distances. In addition to that, aquatic habitats have very varied environmental conditions, ranging from small trickles of streams to larger rivers with cascading ripples and still water pools, to large rivers that have either canyon-bound or large floodplains. So across even relatively short spatial distances, there can be massive landscape distances between habitats, so lots of variability. And this leads to aquatic habitats being among some of the most biodiverse habitats in the world. And particularly, there's a lot of diversity in aquatic invertebrates, which is going to be the focus of the biodiversity that I have been studying. So aquatic invertebrates, there's thousands of different species across broad taxonomic groups. They also, within each species, they have complex life history traits. So many aquatic insects, for example, have an aquatic larval phase where they live in the water, and then they'll emerge as terrestrial adults that can either crawl or fly around between both along the river network or between across the terrestrial landscape. Additionally, aquatic invertebrates had a really important role in the food web, where they are an important source of secondary productivity that links primary photosynthetic productivity to higher trophic levels, such as fish and birds and bats. Also, because they emerge from the river, they're an important link between the aquatic and terrestrial ecosystems. So all these different landscape factors and distances influence the distribution of biodiversity across the landscape. So why we find certain species in certain places. These processes occur through meta-community processes at multiple scales. In short, meta-communities are just sets of interacting communities, essentially what we find in different habitats. And so at local scales, processes related to environmental variation, such as differences in temperature, elevation, flows, and so on, cause species to sort into optimal habitats, supporting different biodiversity in different places. Additionally, if we look at regional scale processes, these differences in dispersal ability and landscape connectivity influence the sharing of species between different communities. So some paths that can disperse over land, others follow the river network, and others can't disperse very well between our communities either. And this creates a nested pattern of biodiversity where different uneven dispersal abilities among community members leads to the differences in diversity. And so to look at these landscape scale processes, we can use parallel models of communities and populations on the landscape to kind of empirically study how the landscape structures biodiversity. So when the landscape is not very well connected and there's a lot of isolation, we would expect to see higher environmental differences in our communities and populations, seeing the local dynamics of meta-communities and patterns of genetic drift among population genetic differences. At more intermediate levels of connectivity, we could have either connectivity along the river network flowing down and then back up between habitats, or we could have intermediate connectivity across terrestrial pathways, so leaving the river and going to another habitat. And here we would expect to find some regional dynamics of meta-community structure and isolation by distance patterns of population genetics. And then at the other extreme, with there's widespread connectivity, insects or invertebrates can get anywhere they want in the network, we would have pretty similar communities, and we would expect to find patterns of gene flow among population genetic relationships. So all of these, to study the influence of the landscape on aquatic invertebrates, like Dave said, I focused specifically on the lower Colorado River ecosystem with a particular focus in the Grand Canyon. So in addition to this just being an amazing place to go and do field work, it's also a very important aquatic ecosystem. It's a limited source of water in an otherwise arid landscape. So it's very important for the biodiversity. It's also very important for the 30 million or so people that live in the desert Southwest who rely on the river for water and power. Throughout the lower Colorado River Basin and in Grand Canyon in particular, there are complex environmental characteristics and spatial relationships that provide a lot of variation in the landscape that we can study patterns of biodiversity. So there's a harsh terrestrial landscape that these rivers flow through because a lot of these rivers are deeply embedded in canyons that can restrict terrestrial connectivity. There's also heavy anthropogenic impacts, such as the Glen Canyon Dam looming large about 15 miles upstream from the beginning of the Grand Canyon segment. And we find extreme environmental gradients, ranging from at high elevations, pine forests, to lush riparian habitats along the rivers, some very steep canyon-bound waterways, and then lower down Sonoran Desert ecosystems. So within the Colorado River itself, the Glen Canyon Dam and other dams in the system highly alter the environmental characteristics in the river. So the dam reduces annual flow variability, so there's no longer the natural spring snowmelt and then seasonal monsoons in the Mainstem River. And it also has the effect of creating artificially cold, clear, and nutrient-poor water in the segments downstream from the dams. And this has the effect of greatly reducing the aquatic invertebrate diversity in the Mainstem River directly below dams. Below the Glen Canyon Dam, only 12 genera of aquatic invertebrates are found, only 3 of these being insects. And this is significantly lower than even other major hydroelectric dams in along the Colorado River. So the Glen Canyon Dam particularly is detrimental to aquatic invertebrates because the harsh landscape causes the impacts of the dam to propagate very far downstream. This brings up the big management question that we were hoping to address with this research. Do the tributaries and other aquatic habitats in this system compensate for the lack of invertebrates that we see below these major dams? So are the tributaries acting as sources of biodiversity that can recolonize what's been lost below the dams? So in Grand Canyon, there's several dozen perennial tributaries that flow into the Colorado River, although pretty much all of these are significantly lower in flow and have much more varied and different environmental characteristics than the main stem river. Also, all of these tributaries are unregulated, so there's no dams or other human alterations to these tributaries for the most part. Additionally, the lower Colorado River Basin, the Grand Canyon is just one segment of this larger river basin that drains a large portion of the arid American Southwest. So there's other dams along the river, such as the Hoover Dam, which I'm sure most of you are familiar with, near Las Vegas. There's then several more dams in this downstream reach of the Colorado River here. There's also the Gila River and the Salt River that flow through like the Phoenix area and are heavily altered by human development. And then there's the Verde River and its headwater tributaries draining the Mogollon Rim, which are, a lot of this is protected, but, and kind of offers a good parallel to what we see in the Grand Canyon, or at least as close as we can get to the unique characteristics of the Grand Canyon. So to understand this landscape scale biodiversity, it's going to require extensive sampling of communities and populations. To understand what's going on across the entire landscape, we have to sample all over the landscape. And so to do this, I've used environmental DNA metabarcoding, which is a method for extracting and sequencing the extraorganismal DNA that's just shed into the water to identify the biodiversity that's in the water without direct observations or collections. And so for this, we rely on only a single liter of water for each sample, plus replication, obviously. And this allows for much more efficient sampling at each location. And because of that, we can use the amount of effort during fieldwork to sample many, many more habitats to provide a broader landscape representation. So I'm going to be using pretty much the same methods throughout all the data I'm going to present in this talk. So I'm going to run through the eDNA methods at the start so you can understand what I've been doing out in the field and how this data that I'm going to present was generated. So in the field, we collect 1 liter of water samples in replicate, and we filter the DNA out of the water and preserve this in the field. So spent a lot of time pumping water through filters in the Grand Canyon. You can see me and Angelica doing right here. Then once we get back to lab, we extract the DNA and we amplify the DNA that we find in the water using a primer set that is specifically targets freshwater macroinvertebrates. So every living thing's shedding DNA, but we're only targeting the invertebrate DNA that we're interested in studying the biodiversity of. This was one of those papers that Dave mentioned that made us say, wow, this is actually something that we can apply to our system in the Grand Canyon. And then once we amplify the DNA, we then do Illumina high throughput sequencing to generate millions and millions of DNA sequence reads of our aquatic invertebrate DNA. And then once we get that sequencing data back, we perform some sequence cleanup, and then all the sequences that are generated are clustered into operational taxonomic units, which are essentially 3% clustering, analogous to species level differences. And so from the DNA, at first we're not able to tell what species they are. We're just detecting unique genetic sequences. But then once we cluster into this species level variability or species level designations, we can then compare to reference sequence databases to identify the closest taxonomic match, and then we can perform our community analysis. Because operational taxonomic unit are not always taking species, I'm going to try and use the term taxa to describe discrete units of biodiversity for the rest of this talk. And our eDNA methods captured a large amount of biodiversity in this lower Colorado River ecosystem. So we detected 1,445 unique taxa that we were at least able to identify to the level of order. And many of these were classified to the insect order Diptera, so little black-fly midge sort of things. Although we did get a very broad representation of all the invertebrate diversity that we were hoping for. And This method captured much higher diversity than comparable studies that have looked in the Colorado River ecosystem and tributaries in the river itself and in some of the headwater tributaries along the Mogollon Rim. So roughly 4 times the amount of biodiversity is being captured by DNA than by traditional morphological identification. So now that I've explained that we detect large amounts of diversity, how is that diversity being distributed across the landscape? How is the landscape at regional and local scales influencing that? So to start, I'm going to talk about spatial distances throughout the lower Colorado River Basin. Specifically, how do aquatic invertebrate communities vary across this large spatial extent? So throughout the lower Colorado River Basin, we took eDNA samples from 70 sites in this spring of 2022, with a high sampling effort in Grand Canyon, and then also throughout all these other reaches of the network, including other spots on the Colorado River, the heavily human-altered reaches of the Salt and the Gila, and then the Verde and its headwaters in the Mogollon Rim. Mogollon Rim, that's about the area of Sedona, if anybody's familiar with Arizona geography. And so we found that there are significant differences in the level of diversity that we find in each of these different regions. So unsurprisingly, lower diversity in the Colorado River, where there's these impacts of the dam that are restricting invertebrate communities. And we find much higher diversity in the Mogollon Rim headwaters, in the Verde River, and then also in the Grand Canyon tributaries, and interestingly, also in the Salt and the Gila River, which, despite their heavy human impact, still have a lot more diversity in them than the Colorado River itself. Although this is just saying that the communities have different numbers of species, it doesn't necessarily tell us how they're related to one another across the landscape. To do this, I clustered the communities based on a hierarchical clustering algorithm that essentially takes similar communities and clumps them closer together and more dissimilar communities further apart. And I clustered into five different groups with the hypothesis that these would roughly match these predefined regions that I outlined in the previous 2 slides. And we found that for the most part, a lot of the communities that we looked at roughly matched with spatially similar habitats. So along similar portions of the network, there's similar communities, and across larger distances, we start to see different delineations. Interestingly, when we look at the Grand Canyon and the Verde River and its headwaters, we see that there's this cluster of purple sites in the draining high elevation wilderness canyons of the Mogollon Rim and some of the inner gorge habitats in the Colorado River. So these are high elevation, high gradient streams with lots of snowmelt runoff, and we see similar communities in these. And noticeably, they're relatively closely spaced across terrestrial distance. This is still several hundred kilometers, but compared to the distance you'd have to go along the river network, they're relatively similarly spaced, indicating that there's either terrestrial connectivity between these habitats or their similar geographic location drives similar environmental characteristics that causes similar communities. So a takeaway from this analysis of spatial distance, we find unique communities that are defined by broad geographic characteristics like their drainage region and terrestrial connectivity. Although you're probably thinking, I was just saying that spatial distance causes different communities. What about in the Grand Canyon? We have these communities that are clearly distinct at very short distances, nearly overlapping, at least how it looks on this map. So to address those community dissimilarities in the Grand Canyon, I'm next going to talk about environmental distance. So the variability in habitat characteristics, regardless of spatial scale. And I'm going to specifically address the questions, how do invertebrate communities differ across tributary confluences? And how do invertebrate communities differ between tributaries? So within the Grand Canyon, there are we can boil it down to two major classifications of aquatic habitats. There's the main stem Colorado River with high discharge, a central location in the river network, and the high human impacts due to the dam that have reduced aquatic invertebrate diversity. On the other hand, we have our several dozen tributary streams that have lower flows, higher elevational gradients, greater flow variability because they're not impacted by dams or much human alteration, so they have the typical spring snowmelt runoff and seasonal monsoon floods that create a lot of variability and stochasticity in the system. And they have very varied aquatic and riparian environmental characteristics that make all these tributaries pretty distinct from one another. And so where these two habitat types meet are at tributary confluences, which are locations of direct hydrological connectivity where the tributaries flow into the main stem river, but they're also locations of high environmental heterogeneity for aquatic ecosystems. So across these very short habitat distances, these very short spatial distances, we have drastic differences in environmental characteristics. So to illustrate, oh, sorry, I had myself there. So there's a few different potential effects that this could have on the communities we find in the tributaries and in the main stem. If these environmental differences are extreme enough, we could find local environmental filtering where from the regional species pool, distinct taxa are sorted into distinct habitats. Alternatively, we could have regional dispersal from the main stem from the tributaries into the main stem, where taxa drift down or disperse out of the tributaries and can recolonize the main stem. This would be great if the dam is reducing invertebrate diversity. This would allow that to recolonize the main stem. Alternatively, we could have the opposite, where there's dispersal from the main stem up into the tributaries, essentially using the tributaries as refuge habitat from inhospitable conditions in the main stem. And so to give you an illustration of the actual study system that we're in, you can pretty clearly see that there are drastic differences from our tributaries into the main stem. So on the left, we have Clear Creek, which flows at, I think, of only about 1 cubic foot per second. And then, boom, you're into the Colorado River flowing at 8 to 12,000 cubic feet per second. And then, even for some of the larger tributaries, such as Tapeats Creek here, which has flows of around 30 to 50 cubic feet per second at base flow, it's still dwarfed by the main stem Colorado River, which it just enters very abruptly, and the habitat environmental conditions change. So in our lab, we like to use the analogy of this would be like riding a bike from a bike path onto an interstate highway. Yeah, it's similar habitat. It's technically similar habitat. It's all aquatic, but they're very different things going on in each. So to actually look at our tributary confluences, we sampled from 21 tributary confluences throughout Grand Canyon and compared the communities we see directly across. So from the blue, we have our tributaries. In orange, we have our main stem. And we compared the differences using measures of community dissimilarity that range from 0 for completely identical communities to up to 1 for completely different communities. We found that for the overall diversity, here we're using Sorensen index, we have very high community dissimilarity of a mean dissimilarity across our tributary confluences of 0.71. Although this just tells us that the tributaries and main stem have different communities, it doesn't tell us how they're different, whether it's the spatial relationships or whether it's the local environmental differences. So to look at that, we can partition our diversity into turnover and nestedness, which are additive components of the overall diversity that can be calculated out separately. And so turnover is the result of local habitat filtering processes where each habitat has its own unique composition due to environmental characteristics. And nestedness is due to differences in regional dispersal between taxa, where some can disperse, some cannot, and this creates nested subsets between our communities. And what we actually found in the Grand Canyon was that turnover was a much larger proportion of the community dissimilarity, with a mean turnover across our tributary confluences of 0.54, while nestedness was mean nestedness was 0.17. So this dominance of turnover among our community dissimilarity indicates that it's local habitat filtering processes that are driving unique communities due to unique environmental characteristics across our tributary confluences. So some takeaways from this analysis of tributary confluences: we find significant discontinuities across these very short spatial distances due to environmental filtering processes. And to address that management question, unfortunately, tributaries do not act as sources of biodiversity for the main stem Colorado River. Instead, the tributaries in the main stem are isolated from one another with distinct communities. So next, I was interested in asking, how do invertebrate communities differ between the tributaries? So if they're different from the main stem that's directly adjacent to them, are each of the tributaries their own unique communities, or are all tributary communities more or less the same? And so to do this, I used a statistical method known as an NMS ordination, which essentially collapses all of the complexity of our community data set into two dimensions, where points that are, each point represents one of our habitats that we sampled, and how close they are in this plot means that is how similar the communities are to one another. So we can see with our main stem, we have very, very tightly clustered communities that are, so they're not very different from one another, whereas our tributaries have a much greater spread. They all have their own unique communities that are different from one another and different from the main stem, which makes sense if we think about the environmental characteristics that we see in these habitats. The main stem Colorado River doesn't change much in characteristics, both its appearance and the flow and the riparian conditions, as well as things like temperature, dissolved oxygen, pH. That doesn't really change much over this entire Grand Canyon segment, spanning almost 300 miles or 450 kilometers. The tributaries, on the other hand, have a lot more variability in environmental characteristics. Some of them are these tiny little trickles flowing through some pretty dense riparian habitat, and also very dense rattlesnakes in Spring Canyon here. Luckily, we didn't see too many of those. And to larger tributary streams with higher flows, but still significantly less than we're seeing in the Grand Canyon. So we have different environmental characteristics that cause different communities in our tributaries. And this was supported by variance partitioning statistical analyses. So we compared the amount of community variance that we observed due to spatial differences, environmental differences, and shared spatial environmental distances that couldn't be isolated from one another. And we find that although spatial and environmental distances are both significant influences on community structure, the environmental difference is a much larger proportion of the variance we observe. And here, by environmental difference, I calculated this distance based on the temperature, the drainage relief of the basin, the D15 substrate size, and the percent barren terrain of the landscape. Although I do want to point out that the residual variance here, 0.7, is pretty high. This is within the realm of what's normal in river networks. There's a lot of random variability in rivers due to things like flash floods, seasonal weather patterns, and so on, which only further increases the variability in tributaries because they're free-flowing and subject to their own unique flow regimes. That just couldn't be captured by this analysis. So to answer the question of how communities differ between tributaries, we find unique aquatic invertebrate communities primarily due to environmental heterogeneity again, and this creates isolated tributaries that are more similar to an island archipelago than to habitats along a river network. So each of these tributaries is kind of isolated off in its own canyon, separated by this main stem river that the same invertebrates can't live there, and they kind of go off into their own unique communities in each tributary. So we found that tributaries are isolated with unique communities, but this doesn't necessarily tell us whether individual invertebrates are just able to disperse between those. Are they completely isolated where nothing's moving around, or are they, or are there invertebrates that are able to move between these tributary communities? So to address this question, I'm going to look at biological distances. So the difference between the populations that we find in each habitat. Specifically asking, are aquatic invertebrate taxa capable of dispersing between habitats in Grand Canyon? So dispersal is a very important key functional trait that impacts an organism's ability to interact with the landscape and with other organisms, both to find mates and to avoid predation and competition, because it allows these individuals to move between habitats and seek out different conditions. And dispersal ability is species specific. So each species has its own unique dispersal ability due to its morphology, its behavior, its physiology. So some aquatic invertebrates have big, strong wings. They can fly over the place. Some water striders are restricted to walking around on the water. Some never leave the flow and are just kind of swept around in the drift. So dispersal ability varies a lot between species. And in aquatic invertebrates, dispersal is really difficult to observe directly. You can't go and put a radio collar on a beetle or on a water strider or a giant water bug and see where it flies around to. So instead of direct observations of movement, we can instead rely on either genetic relationships between populations or on literature-based trait estimates. So you can go and intensively study a single species, and then there are databases that kind of conglomerate all of those different studies into community estimates, community-wide estimates. So for individual species, using genetic relationships is a very good way to observe dispersal, because in order for genes to be shared between populations, it requires individuals to move between those populations to spread their genes. So if there's high dispersal, there's going to be high gene flow, because it would require individuals to move to those new habitats, share their genes, and if this is happening all the time, all over the place between habitats, that ends up leveling out the genetic variability that we would see between populations. On the other extreme, if there's low dispersal, we would find patterns of genetic drift, where essentially if there's high isolation and there's not much movement of individuals between populations, each population would go off on its own random evolutionary path trajectory, creating high variability regardless of the spatial or environmental relationships between habitats. And then in the middle, if nearby populations are able to disperse between each other, but more distant populations are not, we would expect to find patterns of isolation by distance. And so this framework for addressing dispersal has been used in our lab before, using in a lot of these other landscape genetics papers that Dave alluded to in his introduction. So Philipson et al. Found that Abitus herberti, a hypothesized weak disperser, has across all geographic distances on this x-axis, so ranging from comparisons of barely any distance up to 100 kilometers, high variability in FST, which is a measure of genetic variability. So the higher the FST, the more dissimilar the communities are, the more variable they are. Populations are. And so this weak disperser has high variability regardless of geographic distance. This moderate dispersing stonefly has a positive linear relationship where closely spaced populations are similar to one another, and more distantly spaced populations are more dissimilar and have more variability. And then on the other extreme, for a high dispersing diticid beetle, they found low genetic variance across all geographic distances. So even as far as 140 kilometers, there's very little genetic variability in this population. These patterns were also found to hold within the Grand Canyon specifically for a high dispersion caddisfly, low variability across geographic distance along the river network, and a moderate dispersing mayfly with this positive isolation by distance relationship. However, for the hypothesized weak disperser, a water strider that was hypothesized to not be able to disperse very well, they found pattern more akin to high dispersal ability, high panmictic dispersal ability, with low variability across all distances. So in this chart, really, we'd be looking at 3 high dispersers, 2 moderate dispersers, and 1 weak disperser. So if we're trying to compare differences in dispersal ability across the landscape, this would limit our ability to make inferences about what happens for weak dispersers. So to address that limitation of needing to rely on prior hypotheses of dispersal ability, I use environmental DNA-based landscape metagenetics to essentially invert that study design. Instead, looking at the genetic relationships between our taxa and then assigning dispersal ability based on our genetic observations. So to look at population genetics from eDNA, for all the data I've talked about so far, we took our DNA sequences, we clustered them into our species analog operational taxonomic units, and used that to perform our community data. Another method that we could use is to first identify the absolute amplicon sequence variance. So all of the individual sequences that are found in this eDNA data set. And then once we've identified each of these unique sequences, then we cluster into our species analog operational taxonomic units while retaining this intraspecific variation. So retaining the absolute amplicon sequence variance within each of the operational taxonomic units. And from this, we can perform population genetic analyses on all of the taxa that we are finding in our eDNA data. And so, or for all the taxa that have genetic variability within those clusters. And from that, from our data set, out of our about 1,000 taxa, we found 100 that had substantial amounts of genetic variability that allowed us to perform landscape genetic analyses. And like I said, this allows for an inversion of the conventional landscape genetics study design, where we looked at the genetic relationships and then assigned dispersal and thought about what that meant. So to parallel what we found before, for each species, or what I've showed before, for each species, we compared pairwise measures of geographic distance on the x-axis and genetic distance on the y-axis, and plotted a linear regression line through these points to compare whether there's high variance of positive isolation by distance slope or low variance. And because it would be a mess to show scatter plots for all 108 taxa, I'm just going to show the regression lines instead of the scatter plot. So we found that 25% of those 108 taxa showed patterns of moderate dispersal across aquatic pathways. So a positive relationship between river network distance and pairwise genetic distance. We found an additional 24, so 23% of our OTUs with positive relationships between the terrestrial Euclidean geographic distance and pairwise genetic distance, indicating that they have moderate dispersal abilities across terrestrial pathways. Then we found relatively few taxa, only 6.7%, that showed widespread panmictic dispersal with low genetic variance across all distances, only 8 taxa. And then we found nearly half of our taxa, 44%, had patterns of weak dispersal, so high variability in their genetic relationships between populations across all distances, whether those are short or long. So from this, we can you can see that we found a lot of limited dispersal. So kind of mirroring those isolated communities that we find in the tributaries, the populations of invertebrates are also pretty isolated without much direct dispersal, sharing genes between them. And so to look at how this would compare, this direct genetic observation would compare to the literature-based estimates of dispersal, kind of the standard way of assessing dispersal across all members of a community. If we just plugged in the taxa that we found in our eDNA data set to these literature-based databases, we would find that 10 of our taxa we would expect to find low dispersal, 55 we would expect to find moderate dispersal, and 12 we would expect to find strong dispersal. So that did not match what we found with our direct genetics-based observations of dispersal ability. Notably, we found a lot of taxa, 23% of our taxa displayed weak dispersal ability based on direct genetic observations, but would have been classified as moderate dispersers based on the trait databases. So this tells us that most taxa that we found in Grand Canyon displayed weak or moderate dispersal abilities, and that direct genetic observations did not match between literature-based methods and those genetic observations. So there's a few possible explanations for why these genetic-based methods and literature-based methods might not completely match that are related to the landscape. One could be that the taxa that we find in Grand Canyon have different dispersal abilities than predicted by the trait database. So the trait databases tend to be generalized at the level of genus or family to allow them to be broadly applicable throughout continental scales. But in the Grand Canyon, we found a lot of taxa that are found only in the Grand Canyon. Some of these we couldn't even identify all the way to species because there's no genetic description of them. So maybe those taxa have different abilities and even they're close relatives that were used to generate the trait databases. Another possible explanation could be that even if we're looking at the same taxa that are found elsewhere, maybe the landscape structure of the Grand Canyon, the big differences between the tributaries and the main stem and these harsh terrestrial landscape with big canyon walls might limit dispersal ability much more so than will be found in other habitats that support the same species. So to wrap up, like I said, we looked at 3 different types of spatial distances. When looking at spatial distances, we found unique invertebrate communities throughout the lower Colorado River Basin that are roughly defined by drainage region and terrestrial connectivity. When we zoom in to look at environmental distances between habitats, we found some pretty stark ecological discontinuities across tributary confluences, even at short spatial scales. And then looking at actual realized dispersal, so biological distances between populations, we found restricted dispersal in the Grand Canyon. All of this pointing to pretty high isolation of aquatic invertebrates in this ecosystem. So wrapping up some key takeaways, the Colorado River ecosystem supports diverse aquatic invertebrate communities due to the differences in spatial and environmental characteristics between habitats. And this leads to isolated populations and communities. Related to the dam upstream of the Colorado River, unfortunately, the tributaries the communities in the tributaries are not sufficient to rescue the impacts of the main stem alteration in the Colorado River. So with that, I want to thank my lab, particularly Dave, for this amazing opportunity to do all this research in some pretty amazing places and all of their incredible feedback on everything throughout my time here. This talk and all this research would not have been nearly as good or as much fun to do without them. I want to give a particular shout out to Dr. Angelica Kurthen and Aaron Clarenbach, Master of Science, who both defended their dissertations earlier in this week. So it's been a busy week for Dave the last 7 days. I also want to thank my committee, especially Molly Burke, for letting me do all of the genetic like lab work that I did with eDNA in her lab space. And I want to give a special shout out to the USGS Grand Canyon Monitoring and Research Center for taking us down the Grand Canyon with them and showing us this system. I also want to thank my funding sources, Oregon State University, the U.S. Geological Survey, and the U.S. Forest Service. Yeah, so now for the fun part. I want to thank everybody who I've ever met. I want to thank all of the great people in our lab for most of these pictures are from field work or from conferences because that's really where our lab shines. It's been a pleasure to be able to do field work with all of you, go to some amazing places that I never thought I would be able to experience, let alone experience with such great people. And it's really just been a great supportive community. I also want to thank all of the great people who from the USGS and other groups who were part of our field work. We especially shout out to Chris Hunsaker and all the other boatmen who brought us down the river safely, well-fed, and having a lot of fun along the way. And to all the other amazing scientists who were doing research along these trips. When we were on the river, we were doing pretty much 24/7 science, although sometimes there was a lot of waiting around for other people to do their science as well. And yeah, I just feel really grateful that all of this time off out in the wilderness with a group of people I never met before was really just an amazing experience and made friends for life. I also want to thank all of my friends from OSU that I made in grad school. It's been a great community and lots of support, lots of fun, lots of time up in the mountains, which seems to be where everybody spends all of their time unless we're in lab or at field work. I also want to thank all of my friends from Eugene who made the trip up here to see me give this talk. Yeah, it's been great to have such a great group of friends to both support me along in the research and keep me grounded outside of the research. Also, a shout out to all of my friends who are, or I guess I want to say, all these people are also in PhD programs. So they're also, it's great to have people who are going through similar experiences as well. But I want to thank all of my friends from college and that I've made since moving to Oregon who are not involved in science for reminding me that there's more to life, such as running, playing Settlers for the Baton, and really not much else other than those three things. And I want to thank the DiNardos, Acadia's family, for welcoming me into their family and taking me up to the mountains every time they've come out to visit Oregon. It's been great to really feel accepted by you, all of you. And yeah, really a big shout out to my family. They've been supportive my entire life, as far back as I can remember. Surprisingly, this might have been the last time I was in a boat from then until I went to the Grand Canyon. We were not river people. I was not a river person before this research. But they've really just been there for me my entire life, and it wouldn't, yeah, don't know where it would be without them. And I especially want to give a big shout out to Acadia, my fiance, for really just being there alongside me the whole time. Sorry, I'm Dr. DiNardo. She also defended her PhD 2 weeks ago. And so until I have the doctor after my name, I have to refer to her as doctor until then. And yeah, it's just been great starting our family with us and our dog Whiskey. It's just always been such a great time, even through the highs and lows of both of us trying to defend a PhD within 2 weeks of each other. So yeah, let's hope that soon I can be popping the champagne just like you did a few weeks ago. And with that, I will take any questions. Yeah. Yeah, we're good.
Explain how DNA works, like, what exactly, like, what types of DNA sequences are like,
Yeah.
you know?
Yeah, so well, pretty much everywhere, but especially in aquatic systems, everything that's living is constantly giving off DNA. They're shedding cells, they're excreting, they're being eaten and burst open by predators. And so all of that like metabolic activity causes cells to be shed, and then those cells break open. And so DNA is a pretty stable molecule, all things considered. So that DNA will stay in the water column, or in other places, people can take DNA out of the air, out of soil, all over. And then, so the original like sample that I'm taking in the field has DNA from pretty much everything. And then I use PCR primers that specifically amplify aquatic invertebrate DNA. So we use primers that were published by another lab that went through all of the hard trial and error to get that to work. And so when we amplify the DNA, we're only getting the DNA for the most part that belongs to aquatic invertebrates. So that's how we go from all the DNA that's from anything in the water to specifically the portion of the biodiversity we're interested in our studies. Yeah.
Seems like you were looking at like presences and genetic similarities among organisms. Is looking at the relative abundance of different taxa a possibility with this method? Would that be useful for your analysis?
It would be useful. It's one of the biggest like open questions with eDNA as a method is how much can you trust the relative differences in reads and like the reads you get from sequencing. So for most of the community data, I was just using presence absence because we were still getting large amounts of data that are like good to use for the community analyses. But due to both uneven shedding of DNA in the ecosystem and differences in PCR, both primer specificity and PCR stochasticity, you can get differences in the amount of reads that may or may not be directly related. And since we're not looking at individuals, it's hard to get those measures. For the landscape genetics, when I was looking at the population genetics, I used a kind of rank abundance method to get rough estimates of abundance to provide a bit more insight into their population genetics. But there I was just ranking based on the kind of I think 5 different bins of like the bottom 20%, the top 20%, and in between of the read proportions. So if there was one sequence that spiked way high, that wouldn't influence the findings that much there. So, yeah. Yeah. So.
All of your talk about dispersal ability, especially comparing it to kind of the projected metrics, was based off of, or at least thought of based off of their ability. Is there any chance that with all you're talking about the environmental distances, that it's not so much their ability to travel, but that they might have nowhere else to go for their ecological niches?
Yeah, it's a good question. So at like a ecological evolutionary scale, dispersal is more than just the movement of an individual from one place to another. It also is the like decision to leave one habitat and then the ability to successfully establish in another habitat. So for kind of for evolutionary scales and using these genetics methods, we're really asking, are they able to move to a new habitat? And then once they get there, are they able to become a part of the population they're joining and reproduce there? So we're not necessarily going to detect just something flying off and then dying and not really having much impact other than maybe getting eaten by one bird or one fish. It's really whether these populations are connected in a broader ecological evolutionary sense.
Well, it's about time for us to, was there one more question?
One more question.
Okay, one more question.
Thank you.
And then I'll wrap it up.
In terms of your biological diversity and the variability of it between like tributaries and the main stems, do you think you would see the same trends you did like across different seasons, or have you looked at it like on a year-to-year basis and see how that variability from the change?
Yeah, so I looked at this, we took samples in April for 4 different years, so 2021 through 2024. We found very similar patterns year to year, especially on the broad community scale where 1 or 2 species changing doesn't impact that as much. In terms of seasonal variability, I'd imagine there's some amount of variability, especially in streams where there's been a flash flood that past summer in the monsoon season. If we went in the fall, we would find reductions in certain taxa, increases in others based on their ability to respond to those seasonal changes. But we find that over multiple years, those changes kind of ebbs and flows seasonality are relatively consistent between year to year.
Okay, so we're gonna now pivot toward the closed door part of this defense of the committee. And before we do that, I want to announce that we're gonna have a celebration, no matter how that goes. We're gonna have a celebration at the Lilo Dallas residence, because we have a lot to celebrate in the lab with other defenses. And with that, let's thank everyone.